Hello everyone! Do you know what protein is? In this video, you will see an explanation about protein. First of all, I want to introduce the names of our group members. First, there is Jelviona as the reader of slides 4 to 8. The second one, there is Diva Zahra as the reader of slides 9 to 23. The third one, there is Luthfiyah Astried as the reader of slides 24 to 28. And the last one, there is Meviana Arahman as the reader of slides 29 to 47. The contents of this presentation. Here's what you'll find in this presentation. 1. The definition of protein. 2. Types of protein source. 3. The function of protein. 4. Protein digestion process. 5. Protein metabolism in human body. 6. Biosynthesis of protein. So, without any further ado, let's get started!
Definition of protein. Definition of protein. Protein is a polypeptide compound where the polypeptide compound is composed of peptide. These peptides are the building unit of protein, but this peptide is also composed of amino acid. So it can be concluded that amino acids are the smallest compounds that make up protein. The building block of protein are carbon, hydrogen, oxygen, and nitrogen. Protein source. First, vegan protein. Protein that comes from plant. We can get it from soy milk, tofu, mushroom, fish, etc. Second, animal protein. Protein that comes from animal. We can get it from meat, fish, chicken, egg, etc.
And now, I will explain you about the function of protein. For the first is antibody. Antibodies bind to specific foreign particles such as viruses and bacteria to help protect the body. For example is immunoglobulin G. The second is enzyme. Enzymes carry out almost all of the thousands of chemical reactions that take place in cells. They also assist with the formation of new molecules by reading the genetic information stored in DNA. For example is phenylalanine hydrolase. The third is messenger. Messenger proteins, such as some types of hormones, transmit signals to coordinate biological processes between different cells, tissues, and organs. For example is growth hormone. The fourth is structure component. These proteins provide structure and support for cells. On a larger scale, they also allow the body to move. For example is actin. And the last is transport or storage. These proteins bind and carry atoms and small molecules within cells and throughout the body. For example is ferritin. And next, I will explain you about the process of digesting protein in the human body. From the mouth to the stomach. The first step is to digest food by chewing from large pieces into small pieces, which are assisted by the salivary glands and then into the esophagus. After entering into the stomach will release gastric juices containing hydrochloric acid and the enzyme pepsin, which start the breakdown of protein. The acidity of the stomach facilitates the unfolding of the proteins that still retain part of their three-dimensional structures after cooking and helps break down the protein aggregates formed during cooking. The powerful mechanical stomach contraction churn the partially digested protein into a more uniform mixture called chyme. Pepsin, which is secreted by the cells that line the stomach, dismantles the protein chains into smaller and smaller fragments. Protein digestion into the stomach takes a longer time than carbohydrate digestion, but a shorter time than fat digestion. Next is from the stomach to the small intestine. The stomach empties the chyme containing the broken down food pieces into the small intestine, where the majority of protein digestion occurs. The pancreas secretes digestive juice that contains more enzymes that further break down the protein fragments. The two major pancreatic enzymes that digest proteins are chymotrypsin and trypsin. The cells that line the small intestine release additional enzymes that finally break apart the smaller protein fragments into the individual amino acids. The muscle contraction of the small intestine makes and propel the digested proteins to the absorption sites. The goal of the digestive process is to break the protein into dipeptides and amino acids for absorption. In the lower part of the small intestine, the amino acids are transported from the intestinal lumen through the intestinal cells to the blood. This movement of individual amino acids requires special transport proteins and the cellular energy molecule, adenosine triphosphate or ATP. Once the amino acids are in the blood, they are transported to the liver. As with another macronutrients, the liver is the checkpoint for amino acid distribution and any further breakdown of amino acids, which is very minimal. Because ammonia is toxic, the liver transforms it into urea, which is then transported to the kidney and excreted in the urine. Urea is a molecule that contains two nitrogens and is highly soluble in the water. Because amino acids are building blocks that the body reserves in order to synthesize other proteins, more than 19% of the protein ingested doesn't get broken down further than the amino acids monomers. Next is duodenum or proximal jejunum. In adults, essentially all protein is absorbed as tripeptides, dipeptides or amino acids, and this process occurs in the duodenum or proximal jejunum of the small intestine. The peptides or amino acids pass through the interstitial brush border by facilitative diffusion or active transport. Active transport sodium and ATP to actively transport the molecule through the cell membrane. The R group determines the type of transport used. Once passed through the membrane, the amino acids or peptides are released into the intestinal bloodstream and are transported to the liver by the hepatic liver portal vein. This is known as the enterohepatic circulation. In the liver, In the liver, 50 to 65% remain and are used to synthesize protein, nitrogen-containing compounds, and from purine or pyrimidine bases. In some cases, they may be converted to energy. The liver regulates the amino acids levels in the blood. The amino acids that don't stay in the liver pass through and are transported to the rest of the body to be taken up and utilized by other cells. Most branched-chain amino acids pass through the liver. Amino acids are recycled. Just as some plastics can be recycled to make a new product, amino acids are recycled to make a new proteins. All cells in the body constantly break down proteins and build new ones, a process referred to as protein turnover. Every day, over 2,050 grams of protein in your body are dismantled, and 250 grams of new protein are built. To form these new proteins, amino acids from food and those from protein destruction are placed into a pool. So it is not a linear pool. When an amino acid is required to build another protein, it can be acquired from the additional amino acids that exist within the body. Amino acids are used not only to build protein, but also to build other biological molecules containing nitrogen, such as DNA and RNA, and to some extent to produce energy. It is critical to maintaining amino acid levels within the cellular pool by consuming high-quality proteins in the diet, or the amino acids needed for building new proteins will be obtained by increasing protein destruction from other tissues within the body, especially muscle. This amino acid pool is less than 1% of total body protein content. Thus, the body doesn't store protein as it does with carbohydrates, as glycogen in the muscles and liver, and lipids as triglycerides in adipose tissue. And here is an overall picture of the process of digesting protein in the human body.
Protein metabolism. First, the protein found in our food is digested in the stomach and the gut becomes AA. Second, absorbed and carried by the blood to the liver. Third, a portion of the AA is taken by the liver. Protein in body cells is formed from amino acids. Fourth, if there is an excess of AA from the amount used for protein biosynthesis, then the excess AA will be converted into keto acid, which can enter the citric acid cycle or be converted into urea. Fifth, the liver is an organ in the body where catabolic and anabolic reaction occur. Reaction of amino acids metabolism. The initial stage of the AA metabolic reaction involves the release of the AA group through two processes, namely is transamination and deamination. Transamination is an AA catabolic process that involves the removal or release of amino NH2 from one AA to another AA, and this is the form of the reaction. In this transamination reaction, the NH2 group release is achieved by a keto acids, so that new AA is alanine, glutamic acids, and other keto acids are formed. The transamination reaction is reversible. In this reaction, there is no missing amino group because the amino released by AA are achieved by keto acids. This transamination reaction occurs in the mitochondria and cytoplasmic fluid. Oxidative deamination. In the deamination reaction, the NH2 group is released in the form of ammonia, which is then excreted from the body in the form of urea in the urine. High levels of ammonia are toxic to the human body. AA with transamination reaction, glutamic acids. In some cells, for example, bacteria, glutamic acids can undergo an oxidative deamination process that uses glutamate dehydrogenase as a catalyst. Glutamic acids plus NAD plus beta-glutaric acids plus NH4 plus NADH plus H plus. In this process, glutamic acids release an amino group in the form of NH4. Urea cycle related to the deamination reaction, which released NH2 in the form of ammonia, which is released by the body in the form of urea. Hanscraft and Urt Henselage 1932 suggested a series of chemical reactions regarding the formation of urea. They argue that urea is formed from ammonia and carbon dioxide through a series of chemical reactions in the form of cycle. Urea cycle. First, the formation of urea mainly takes place in the liver. Second, urea is water-soluble compound, neutral in nature, found in urine which is excreted from the body.
Now, I'm gonna explain you about biosynthesis protein. Protein biosynthesis is the process in which cells build proteins. The term is sometimes used to refer only to protein translation, but more often it refers to a multi-step process, beginning with amino acid synthesis and transcription, which are then used for translation. The protein synthesis process is divided into two stages. The first one is transcription, which is the process that will produce mRNA with DNA as the template. The second one, there is a translation, where the mRNA produced from the transcription process will function as a template at this stage, and the printout is called a protein. Transcription. RNA polymerase is the enzyme responsible for making mRNA copies of genes. DNA unzips at the site of the gene that is needed. RNA polymerase matches bases in the sense that with RNA bases, building a strand of mRNA that carries the information encoded in the DNA. Encoded in DNA is a signal telling RNA polymerase where to stop. Transcription ends at that point. The completed mRNA molecules then moves from the nucleus to the rogue endoplasmic reticulum for translation. This is an overview about the process of transcription. Translation. Initiation begins with a tRNA bearing methionine attaching to one of the ribosomal units. The codon for methionine is a universal start codon for reading the mRNA strand. The ribosomal unit binds to mRNA where the code for met is located. The anticodon UAC of the tRNA matches the start codon of mRNA AUG. The larger ribosomal subunit now binds to the smaller unit, forming a ribosomal complex. The tRNA binds to the first active site on the ribosome. Translation may now begin. A new peptide bond forms on the catalytic site. The process continues until the ribosome finds a stop codon. The subunit detect from one another. The mRNA is released, and the polypeptide chain moves down the endoplasmic reticulum for further processing. The initial met is removed, and the chain is folded into its final shape. However, this polypeptide can be used. Completion of protein, namely after the translation process, occurs in the Golgi body. The refinement process that occurs in the Golgi body is the folding process, acetylation, methylation, and carboxylation. After the protein is complete, it will be stored in the lysosome and ready to use. DNA replication. DNA replication is the process by which DNA makes a copy of itself during cell division. Replication enables the formation of two identical DNA molecules from a single double-stranded DNA. There are three theoretical ways of DNA replication. The first one, there is conservative. All of the original DNA spiral ladders are retained and will serve order for new spiral ladders. The second one, there is semi-conservative. In semi-conservative replication, the spiral ladder open first so that the two polynucleotide strands will separate from each other. However, each of these strands is retained and acts as a template for the other for new polynucleotide strands. And the last one is dispersive. Both polynucleotide strands undergo fragmentation in a number of places. Then, the polynucleotide fragments that are formed will become molds for new nucleotide fragments so that the old and the new fragments will be found alternately in the new spiral staircase. The mechanism. The first step in DNA replication is to unzip the double helix structure of the DNA molecule. This is carried out by an enzyme called helicase, which breaks the hydrogen bonds holding the complementary bases of DNA together, A with T and C with G. The separation of two single strands of DNA creates a Y shape called a replication fork. The two separated strands will act as templates for making the new strand of DNA. One of the strands is oriented in the 3 to 5 direction. This is called the leading strand. The other strand is oriented in the 5 to 3 direction. This is called the lagging strand. As the result of their different orientation, the two strands are replicated differently. On the leading strand, a short piece of RNA called a primer, produced by an enzyme called primase, comes along and binds to the end of the leading strand. The primer acts as the starting point for DNA synthesis. DNA polymerase binds to the leading strand and then walks along it, adding new complementary nucleotide base A, C, G, and T to the strand of DNA in the 5 to 3 direction. This sort of replication is called continuous. On the lagging strand, numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. Chunks of DNA called Okazaki fragments are added to the lagging strand, also in the 5 to 3 direction. This type of replication is called discontinuous, as the Okazaki fragments will need to be joined up later. This is an overview about the process of Once all of the bases are matched up, A with T and C with G, an enzyme called exonuclease strips away the primers. The gaps where the primers were are then filled by yet more complementary nucleotides. The new strand is proofread to make sure there are no mistakes in the new DNA sequence. Finally, an enzyme called DNA ligase seals up the sequence of DNA into two continuous double strands. The result of DNA replication is two DNA molecules consisting of one new and one old chain of nucleotides. This is why DNA replication is described as a semi-conservative. Half of the chain is a part of the original DNA molecule. Half is a brand new. Following replication, the new DNA automatically winds up into a double helix. This is the references that used in this video. Thank you for watching. If you have any question, please write down on the comment section. Don't forget to like and share this informative video. And goodbye.